Any questions? Just out of curiosity, since I posted the lecture notes, did anyone look over those prior to class? Well, I guess I don't have to give you an optional point opportunity then. Just going to give an optional point opportunity if you look at the notes. Everybody see the note up here about making sure your cultures are either started today or out? If you made a culture on Friday, make sure you refrigerate it. If not, you need to make one today. When we talked about microbial growth, we talked about the fact that organisms obviously need food resources in order to sustain that growth. So what we need to get into now and spend some time on, and this will be very relevant to things we talk about in lab later on, particularly after the first lab exam, is the whole issue of microbial metabolism. One of the concepts that I have pushed quite a bit so far is the fact that prokaryotes, even though they are very simple structurally, have a very sophisticated metabolic capacity. That makes things a little bit more difficult when we start talking about concepts of metabolism than they were in cell biology, because we talk primarily about eukaryotic metabolism, and that, for all its complexity, is much more straightforward than microbial metabolism. When we talk about metabolism, remember we're talking about the totality of all of the chemical reactions that take place within the cytoplasm of the cell. And ultimately, remember there are two major categories of metabolic reactions. Remember what those two categories are? Catabolic and anabolic. Okay, what do catabolic reactions do? What's their purpose or intent? Break down complex molecules to simpler molecules for what purpose? Make ATP, okay. To make energy, typically in the form of ATP, the universal currency of the cell that it uses to do work. What is the anabolic role of metabolism? Well, what is anabolism first? What do anabolic reactions refer to? Biosynthetic reactions, so taking simpler molecules and putting them together to form more complex molecules. So taking amino acids together and assembling them into a polypeptide chain, or synthesizing a lipid from glycerol and fatty acids. While catabolic reactions are typically degradative and energy releasing, and we try to trap that energy in the form of ATP, anabolic reactions are typically energy requiring reactions. They require an input of ATP. Where does that ATP come from? It comes from the catabolic reactions, correct? So there's this relationship between catabolic reactions and anabolic reactions that's really important to understand. Well, basically metabolism is all about moving around electrons. That's what metabolism is. Moving things from electron donors to electron acceptors. In catabolic reactions, the electron donors are typically complex organic molecules like carbohydrates, lipids, proteins, and nucleic acids. And those kind of molecules are typically loaded up with hydrogen. And what is a hydrogen? Well, it's just a proton and electron, right? So if you're moving hydrogens around from one molecule to another, you're moving electrons. So in biological systems, metabolism is primarily about moving hydrogens from one substance to another. They can be taken off of a food molecule like sugar. They can be temporarily transferred to a coenzyme like NAD or FAD, remember those things? And then ultimately, though, those electrons or hydrogens have to be pulled off the coenzymes and taken somewhere else, like donated to an oxygen molecule to make water. So one of the things that we want to do when we talk about metabolism is to talk about where are the electrons coming from? Where are they going? Do they have an intermediate place that picks them up? So what's kind of the electron/hydrogen flow? When we start to categorize metabolic reactions and pathways in prokaryotes, we need to concentrate on two big things. Number one, where are the organisms getting their energy? Where are they getting their food? Where are they getting their source of hydrogen/electrons? They're going to start stripping off of those molecules to generate electron flow and make ATP. So what energy sources are used to make ATP? But we're also going to be interested in where do organisms get their source of carbon? Why do you need carbon for biosynthesis? To make other molecules. I mean, you can't just degrade things if you're going to grow. That'll give you the energy. But if you're going to grow and replace cell parts, so a cell can divide by binary fission, you need to make new membranes, you need to make new DNA, you need to make new proteins. How do you do that? You need sources of carbon or carbon skeletons to be able to synthesize new molecules. So where's that carbon coming from? Okay, we do you a little bit of disservice in introductory courses like cell biology, at least from a microbial standpoint we do. We tend to characterize all organisms into two batches when we talk about metabolism: autotrophs and heterotrophs. And how do we distinguish typically between an autotroph and a heterotroph? What's the difference? Which are we? Are we heterotrophs or autotrophs? We're heterotrophs. We eat others. We get our energy from the organic molecules of other organisms. What are autotrophs? Where do they get their energy? From the sunlight. Okay, so those are the photosynthetic organisms. So we make this kind of distinction. Autotrophs are the producers, heterotrophs are the consumers, right? That's okay. Well, in the microbial world, it is more complicated than that. And the terms autotrophy and heterotrophy mean something different than what we've told you in the past. And so that's one of the things we've got to kind of go back and kind of re-educate you about. This is going to be an important diagram for you. Because what this is going to do is this going to allow us to kind of characterize microorganisms metabolically based upon two things. Where do they get their energy? Up at the top. Okay? Where do they get their carbon? Along the side. In the microbial world, in order to fully characterize an organism, we have to determine both of those factors. Where's its energy coming from? Where is its carbon coming from? There are four possible combinations of organisms then that we have to deal with and understand. The photoautotrophs, the chemoautotrophs, the photoheterotrophs, and the chemoheterotrophs. So this is a little bit different than what we've been telling you before. Okay, we've just been using the terms autotroph and heterotroph before. Producer and consumer. In reality, it is more complicated than that in the microbial world. So, let's take a look at energy sources first. There are two potential places where a microorganism can get its energy. And one is from chemical compounds. These compounds are serving as electron sources. And part of energy acquisition, making ATP, involves taking the electrons off of these compounds and donating them someplace else. Getting electrons moving. Getting electrons being transported. Making ATP by that process. Now, in our reality, what we understand about respiration and metabolism so far is that all we've ever talked about is the fact that the source of energy using chemical compounds is organic molecules. Okay. Like glucose. Okay. When we talk about cellular respiration, we always assume we're starting out with glucose. Now, if you remember things from cell biology, remember that there are a lot of different organic molecules like carbohydrates and lipids and proteins and nucleic acids that ultimately can be converted into glucose or other compounds that are part of the glucose breakdown process. But we always assume kind of organic molecules. Okay? But in the microbial world, another possible source of energy are inorganic molecules. Like hydrogen or nitrate. So in some situations, a distinction has to be made when you're talking about what's your source of energy. Oh, it's chemical compounds. Whether that energy is an organic compound or an inorganic compound. If the source of energy is an organic compound, we typically call those organisms organotrophs. If it is an inorganic source, we call them lithotrophs. This literally means rock eating. They're eating inorganic molecules. Okay, so that's a little confusing. Fortunately for you, on this scheme right up here, there's not that distinction made between organotrophy and lithotrophy. I'll show you another slide in a second where they do use that terminology. I just wanted you to be aware of that. Okay. So, energy sources, chemical compounds, chemo. Another possible source of energy is light energy. If your source of energy is light, then those compounds, those organisms are called phototrophs. So for our purposes right here, energy sources, either chemotrophs or phototrophs. What's your source of carbon? If your source of carbon is inorganic, CO2, then you are said to be an autotroph. If your source of carbon is an organic molecule, then you are said to be a heterotroph. So really, technically speaking, the term autotroph and heterotroph only have to do with where you get your carbon. They don't have anything to do with where you get your energy. So that's where we kind of mislead you a little bit in introductory courses. So there are four possible combinations in this grid. If you put energy sources, two energy sources at the top, and 2 carbon sources on the side. What are the possibilities? One possibility are the photoautotrophs. What does that mean? They get their energy source from sunlight. They get their carbon from inorganic carbon dioxide. What kind of organisms are photoautotrophs? Plants, algae, and then there are several bacteria, including the cyanobacteria that we talked about. They carry out that type of photosynthesis that uses water as a source of electrons and produces oxygen as a byproduct. So they carry out that oxygenic photosynthesis. But then we also talked about, when we did our survey, that there were some kind of unique bacteria that carried out a different type of photosynthesis. They're still photoautotrophs, okay? They're still getting their light energy from the sun and their carbon from inorganic carbon dioxide. How do they differ? Their source of electrons is not water, but it's something like hydrogen sulfide. And when they split that hydrogen sulfide during the photosynthetic reactions, they don't produce oxygen. Hydrogen sulfide doesn't have any oxygen. They produce sulfur instead. So this was the anoxygenic photosynthesis. It doesn't produce O2. It produces things like sulfur instead. Okay, let's go down here for a second. Okay. What if your source of energy is chemical compounds, and your source of carbon also is organic molecules? And what are you? You're a chemoheterotroph. This is what we are. Okay, we're chemoheterotrophs. This is what a vast majority of bacteria are as well, including anything we use in lab, including your unknown. Because where are they getting their chemical compounds to produce energy and their chemical compounds to use as carbon sources? From broth or auger. Okay. Now, we're going to go dig deeper into this chemoheterotrophy in a minute and talk about the differences between different strategies to extract energy from these molecules and to get the carbon skeletons. So things like aerobic respiration, anaerobic respiration, and fermentation. So we'll come back and talk about those later on. So in cell biology and other introductory courses, basically these are the only 2 scenarios we even talk about. Okay? We talk about plants and photosynthesis, and we talk about all other organisms, basically, that degrade organic molecules to survive. And we just shorthand it and call them autotrophs and heterotrophs. But in the microbial world, there are other possibilities and strategies for survival. So, for example, there are organisms that use chemical compounds as their source of energy, but they use carbon dioxide as their source of carbon. So these are chemoautotrophs. Well, that's a very interesting strategy. And it's not one we see anywhere other than the microbial world. The ones that are really interesting are these guys, the bacteria who can use inorganic molecules as their source of energy, and inorganic CO2 as their source of carbon. These can live entirely on inorganic molecules. These can literally, if you take it kind of literally, live on rock. In introductory courses like cell biology, we tell you that all organisms require the sunlight for survival, either directly or indirectly. Either to produce the energy or to produce the organic molecules that other organisms need. These are an exception to that rule. These can survive completely in the absence of solar energy. Why? Because they're using entirely inorganic molecules. Inorganic chemicals as their source of energy, inorganic molecules as their source of carbon. So, about 4 billion years from now, we believe that the sun will expand into a giant red star and basically burn itself out. And the Earth will then be kind of a sunless hulk, a big rock. And we've always told you in introductory courses that all life on Earth will cease because there's no source of solar energy to sustain life. Not exactly. These guys could survive. These guys could survive strictly on inorganic molecules. Here's another strictly microbial strategy. Photoheterotrophy. So what are these guys doing? They're using light as their source of energy. So they're using light to make ATP. But they are using organic molecules as their carbon source. You know, wait a minute, if they've got organic molecules already available as their carbon source, why aren't they using them as energy sources as well? That's a very interesting strategy. Use sunlight to make your energy and use organic carbon as your carbon source. We're going to talk about this strategy just a little bit. These represent some bacteria we haven't looked at yet. They're the green and purple non-sulfur bacteria, as opposed to up here we have the green and purple sulfur bacteria. So this is a different strategy of photosynthesis than this is. Okay, I know this seems confusing, but one of the take-home messages that I do want you to understand is that this clearly illustrates the metabolic diversity of microorganisms compared to eukaryotes. Eukaryotes fall into one of these two categories, this one right here or this one right here. All eukaryotes are one of these two things. These strategies are also well represented in the microbial world. These are found exclusively in the microbial world. Now, what we're going to do is we're going to spend most of our time in metabolism down here, talking about the chemoheterotrophs. This is the most commonly employed strategy by microorganisms. This is the strategy used by your unknowns and any organism we use in lab. Once we dig through this, we'll talk just a little bit more about chemoautotrophy because it's so weird. And then we're going to switch over to the photosynthetic organisms. And we'll do some contrasting between the strategies that the photoautotrophs use versus the strategy that the photoheterotrophs use. There's a really, really interesting bacterium that falls into this particular group. And we'll kind of end the discussion of metabolism with this particular organism. This organism is an organism called the bacteria that will eat anything. And it's really a crazy bacterium in terms of the way it metabolizes. Okay, so we're going to be working down here now. We're going to start here and spend quite a bit of time talking about chemoheterotrophs. This slide up here, don't worry about this right now. This is a good review slide. This only deals with the energy acquisition aspect. So really the top two boxes. And it breaks organisms down into these subcategories, and so you'll see reference to organotrophs and lithotrophs. I think this particular slide will be helpful when you're trying to kind of sort all this stuff out once we've gone through everything, because some of the strategies that we'll talk about will be well summarized in this particular slide. So don't worry about this one right now. There's a lot of information on there. Oh, I didn't know I had this one. What do we got going here? Okay, so this slide is just indicated to show the relationship between heterotrophy and autotrophy. This shows the relationship in terms of carbon flow between those two strategies. I'm going to wait on that one and probably come back. That probably shouldn't be in that particular position. Let's go there. So let's take a look at chemoheterotrophs. And if we're going to be technically correct, I probably should put on this particular slide, these are the chemo-organoheterotrophs. So these are organisms that are using organic molecules as their energy source, as well as organic molecules as their source of carbon. There are three major strategies for obtaining energy if you are a chemoheterotroph. They are aerobic respiration, anaerobic respiration, and fermentation. Another thing that we don't necessarily tell you in cell biology, but kind of imply it, and most people believe it up to this point, is that they believe that these last two terms are synonymous, that anaerobic respiration and fermentation are the same process. Both occur in the absence of oxygen, so they have a similarity. But biochemically, they're very different pathways and they're very different strategies. So I want to make sure you understand the distinction between anaerobic respiration and fermentation. That'll be one of the kind of goals. Which one of these strategies is most efficient in terms of getting the maximum amount of ATP yield out of a molecule of glucose? Aerobic. Okay. Which one is least efficient? Fermentation is actually least efficient. Okay. Fermentation only produces a net of 2 ATP. Do you remember the number for aerobic respiration? Yeah, it's that, there's that fudge factor, 36 to 38, depending upon what happens to the reduced NAD produced during glycolysis. Don't worry about that right now. These two terms, or strategies, have the term respiration in their name. Anytime you see the word respiration, it tells you something important about how that pathway works. If you have a respiratory pathway, there is going to be an electron transport system involved. So that's kind of a requirement for being a type of respiration, is you've got an electron transport system. So am I implying that because fermentation does not have the word respiration in it, that there's no electron transport system in fermentation? Correct, I am. Okay, there's no electron transport chain. Remember what the electron transport chain's utility was. Remember that its purpose was to recycle the coenzymes from the reduced form back to the oxidized form, so glycolysis could keep on going and the Krebs cycle could keep on going. But what happened during that recycling process? Because of the way the electrons were passed through the electron transport chain, remember that we got a hydrogen ion gradient built up on either side of the membrane? And then that hydrogen ion gradient was a source of energy to make ATP. So the electron transport chain is where you get your really big ATP payoff. So you do produce a significant amount of energy by anaerobic respiration because there is an electron transport chain present. But not quite as much as you get in aerobic respiration. So the ranking is most efficient, less efficient, but still pretty decent, pretty inefficient in terms of ATP production. and there's a difference enough in efficiency that you can distinguish anaerobic respiration from aerobic respiration by relative growth on plates that are put in a gas pack versus an incubator. Because what does a gas pack generate? What kind of an environment? It generates an anaerobic environment. Okay? So those organisms that can tolerate the anaerobic environment, remember what we call those? Those were the facultative anaerobes. Grow, but because they don't produce as much energy as they would in the presence of oxygen, grow less in the gas pack than they would in a normal incubator where oxygen is present and they can do aerobic respiration. So what are the three phases of aerobic respiration? What phase does glucose go through first? Glycolysis, then Krebs cycle, or it's also called tricarboxylic acid cycle, and then electron transport. Okay. The electron transport chain, remember, is basically a factory for recycling coenzymes like reduced NAD and reduced FAD. And as those electrons go through the electron transport chain, remember they're transferred from one kind of compound to another, to another, to another. And ultimately to oxygen, where water is formed. What else is formed during electron transport chain besides water? Okay, ATP is formed. What else besides water up here? ROSs. Remember, the reactive oxygen species are formed typically, which is kind of a byproduct of the system. This last compound in the electron transport chain that picks up the electrons and then donates them up to oxygen to make water is called the terminal electron acceptor. And the terminal electron acceptor can vary from species to species. One common terminal electron acceptor is a compound called cytochrome A3. But not all organisms use cytochrome A3 as their terminal electron acceptor. Some use a different compound. So might this be a way we could differentiate between organisms? What's your terminal electron acceptor? Is it cytochrome A3 or is it something else? Yeah. And there is a test you can use called the oxidase test, which gives you a very easily readable, interpretable result if your organism has cytochrome A3 as its terminal electron acceptor. You smear some organism on a little piece of paper containing some chemicals, and it turns blue if you use cytochrome A3. And that's considered to be a positive oxidase test. And we're going to do that test in the lab, and that's going to be a pretty important test for some of you in terms of determination of your unknown, whether your organism is oxidase positive or oxidase negative. Okay? So one little difference in an organism right here, in that very final compound in the electron transport chain, can be used to identify microorganisms. So, what's going on in anaerobic respiration? Is glycolysis occurring? Yep. Is Krebs cycle occurring? Yep. Same as it does in aerobic. Is there an electron transport system? Yep, it's a type of respiration, right? During the electron transport chain, are coenzymes recycled? Yep. Is ATP produced? Yep. So what's the difference? Where do the electrons get picked up, and what do they end up as? In aerobic respiration, by definition, the electron acceptor is oxygen, and the final reduced product is water. In anaerobic respiration, the electron acceptor is something other than oxygen. And what's it turned into? Well, it depends upon what that substance is. For example, it might be inorganic nitrate. And if inorganic nitrate has some electrons added to it, it is reduced into nitrite. For a different organism, it might be sulfate, a sulfur chemical. If that's the case, then the end product is a sulfide. Typically, this electron acceptor is an inorganic molecule, and it's got oxygen in it somewhere. That's typical. So nitrate, nitrite, sulfate, sulfide. Inorganic, but they contain oxygen. Rarely, that electron acceptor down here can be an organic molecule. That gives you some interesting possibilities for things that can be produced by anaerobic respiration. Now, remember that our ATP yield is a little bit less than it is in aerobic. But with few differences, this aerobic and anaerobic respiration is really more similar than they are different. But it's an interesting strategy, because what it allows organisms to do, particularly if they are the facultative organisms, is if oxygen's present, they use it, and they have the most efficient type of energy production there is. But if oxygen becomes unavailable, then they can switch over to this other strategy and use other electron acceptors, sacrifice a little bit of energy production in the process, but they still can survive. And so this makes them more adaptable and more able to survive in environments where oxygen availability might fluctuate or oxygen might become depleted for one reason or another. So again, it shows the kind of metabolic sophistication of microorganisms when you can do this. Okay, so what about fermentation? Is there glycolysis occurring in fermentation? Yep. Is there Krebs cycle? Nope. Is there electron transport system? Nope. If we're starting out with glucose, what is the end product of glycolysis? Do you remember?
Pyruvate.
Pyruvate. Okay. So glycolysis produces pyruvate. And then if oxygen or some other electron acceptor that works with the electron transport chain is present, then we can have respiration occurring. We can either have the aerobic or anaerobic respiration. But in fermentation, we've got a whole different strategy. What happens to that pyruvate?
We make lactic acid.
You can make lactic acid, and that's what our muscle cells do. Why do we make lactic acid? Because we have the enzymes that can convert pyruvate into lactate. Are there microorganisms that can do that? Yep. Okay. The microorganisms that cause milk to sour are lactic acid producing bacteria. And the lactic acid causes the pH of the milk to drop. That causes the milk protein to denature. The milk curdles and gets nasty. Okay. But are there other strategies of fermentation in the microbial world besides lactic acid production? Yes, absolutely. Why are these particular steps necessary to keep the organism alive? Why can't we just stop at pyruvate? Remember? Because pyruvate production involves stripping some hydrogens off of glucose. And where do those hydrogens go? Where are they picked up by? Remember the coenzyme NAD? And NAD becomes reduced. And eventually, you run out of your oxidized NAD, and therefore glycolysis stops. That's not good. So if we're going to make the 2-ATP by glycolysis, we've got to have some way to recycle those reduced NAD coenzymes back to the oxidized state again. Now, in the presence of oxygen or some other compound that supports respiration, the electron transport chain does that, right? But what happens in fermentation? In fermentation, there's got to be something, some step in there that converts pyruvate into a more reduced compound. Where do those hydrogens come from? From the reduced NADs produced during glycolysis. What happens is those reduced NADs donate their hydrogen back to pyruvate. They're recycled again. So fermentation provides the recycling that the electron transport chain does for respiration. Okay. So here's a good summary of those three things: aerobic, anaerobic, fermentation. All have glycolysis. These two have both Krebs cycle and electron transport. Okay. Glycolysis, Krebs cycle are producing reduced coenzymes. Those reduced coenzymes have to be recycled back to their oxidized state again to keep this going. That's what the electron transport chain is doing. Fermentation, glycolysis is producing reduced coenzymes. But there's no electron transport chain to recycle this reduced NAD. So what happens? Another chemical reaction is used instead. Some biochemical process occurs that adds hydrogens to pyruvate that recycles those coenzymes, and the products that are produced are called fermentated products. So lactic acid is just reduced pyruvate. Where did the hydrogens come from? Reduced NAD. Ethanol is reduced pyruvic acid. Where did the hydrogens come from? Reduced NAD. Why do some organisms produce ethanol and others produce lactic acid? Depends on the enzymes that you make. Some organisms have the enzymes that can reduce pyruvate into lactic acid. Some don't. Some have the enzymes that can reduce pyruvate into acetaldehyde and then acetaldehyde into ethanol. Others don't. It's all a matter of your genes. So fermentation is going to be pretty interesting to us. when we start to get information about what is our unknown and how do we differentiate one species from another. Now, we will do these tests over here, okay? And we want to know whether your unknown, for example, is an obligate aerobe, it can only do aerobic respiration, or whether it's facultative, whether it can do both aerobic and anaerobic. We'll want to know that. And we use the gas pack to do that. But there's a whole wealth of information we can determine about how an organism ferments if it has the ability to do that. And a couple of the things that we'll want to know is, number one, in fermentation, what kind of substrates can the organism use in fermentation? Can it ferment glucose? What do you suppose? Think most organisms can ferment glucose? Most can. There are a couple that can't, that have to have other sources of sugars for fermentation. But, for example, it's also interesting to know whether an organism can ferment something like sucrose. Remember what sucrose is? What is sucrose? What's it made of?
Is it glucose and fructose?
It's a disaccharide. It's made of a glucose bonded to a fructose. So if you're going to be able to ferment sucrose, what do you suppose you're going to have to do to that sucrose first? You have to be able to break that bond, right? And then once you break the bond, you can directly use the glucose, or you can convert the fructose into glucose. So, do you have the enzyme sucrase? If you don't, you can't ferment sugar. What is lactose? What kind of a sugar? Monosaccharide or disaccharide? Disaccharide. It is made up of glucose and galactose. Do you remember the enzyme that breaks down lactose into glucose and galactose? What it's called? It's called beta-galactosidase. Remember that from the lac operon? Okay. So if you can't ferment lactose, one of the possibilities is, I don't have the enzyme beta-galactosidase. So seeing what kind of things organisms can ferment is illustrative and tells you something about the enzymes that they have. That's good information to know to start identifying things. Another important thing about fermentation is, well, let's say you can ferment something. What do you produce when you ferment it? We've already mentioned two possible outcomes of glucose fermentation. Lactic acid being produced, or ethanol being produced. Are there other possibilities? You bet. This shows you just some possibilities of fermentation. Pyruvate being at the center of this particular diagram, because that's what glycolysis produces. But look at the different possibility of different types of fermentative byproducts that can be produced. Several acid compounds, including butyric acid, succinic acid, propionic acid. Okay? Butyric acid is really nasty smelling. This is one of the things that's produced when butter gets rancid. Alcohols are produced. Acids are produced. And other types of acids are produced. There's a whole constellation of different types of fermentative byproducts that are produced. So that's what we'll pick up tomorrow. We'll talk a little bit more about fermentation.